Hello, and welcome to this Nature Research custom media webcast titled BioSAXS, a method to accelerate and de-risk antimicrobial drug development. My name is Nikki Forrester, and I will be your moderator. Today's webcast is produced by GenScript. In 2019, the World Health Organization declared antimicrobial resistance to be one of the top 10 global public health threats facing humanity. But low profit margins and a high risk of cross-resistance have slowed advancements in antibiotic research and development. Novel treatments that offer new modes of action combined with a low risk of resistance are urgently needed. But screening compound libraries of novel antimicrobials for new modes of action is still time-consuming and costly. BioSAXS is an emerging method for testing up to thousands of compounds to classify them based on ultrastructural changes that correlate to their modes of action. Today's webcast will discuss how BioSAXS can be used to classify antimicrobial peptides according to their mode of action. We'll begin the webcast with a presentation from Dr. Kai Hilpert from St. George's University of London and Tika Diagnostics. We'll end the webcast with a Q&A. To ask a question, just type it in where it says Ask a Question and then press Submit at any point during the webcast. We'll do our best to answer all of your questions today. And now over to Dr. Hilpert.
Welcome, everybody. It's a great pleasure to be here and presenting to you, and thanks for coming. And today I would like to talk about our new method that we developed based on BioSAXS to accelerate and de-risk antimicrobial drug development. My name is Kai Hilpert, and I'm a reader or associated professor in infection immunity. I'm also one of the founders and directors of Tika Diagnostics. It's a spin-out of St. George's University of London, so I think I forgot to say I'm working at St. George's University of London. Before I start, I would also like to thank my collaborators, which we were working closely together to actually develop this BioSAXS for the purpose of antimicrobial drug development. So first, that is the group of Axel Rosenhahn at the Ruhr University Bochum, and also Ralf Mikurt's group at the Karlsruhe Institute of Technology. I also would like to thank GenScript for sponsoring this event and make it possible that we all come together today. First, I would like to walk you through the content of this talk, which is a very short overview of our research and the motivation why we do it. And then I would like to introduce Cardionic antimicrobial peptides, how we synthesize them, and then focusing on the BioSAXS and that we can measure bacteria and antimicrobial compounds with the BioSAXS. And there will be a take-home message and time for questions. As I said, I'm one of these founders and directors of Tika Diagnostics, and a colleague of mine and myself, we discovered that some peptides can actually stimulate the growth of slow-growing mycobacteria, especially also Mycobacteria tuberculosis for humans, but also mycobacteria that are relevant in animal healthcare. It was quite a journey. It was very interesting and enrichment in my understanding of the academic research versus the more industrial research, and it was great finally then have a product out of the market. The second point, most of my time is basically more about the academic question where we do bioactive peptides, mainly antibacterial peptides and antifungal peptides, and we kind of started a little bit on antibiofilm peptides as well. Together with a colleague here at St. George's, Farron Waldemar, we have a collaboration to investigate anti-cancer peptides, which works really well. And they also did some studies on immunomodulatory peptides. Recently, we have found some wound healing peptides, and we're trying to understand the human lung peptidome. So there's lots of peptides in the lung that are produced directly or as a product of the cleavage. And that obviously also changes by infections. And we're trying to understand the whole dynamic that these peptides bring. So in summary, you basically want to push peptides forward and finish up preclinical development in order then to move into clinical trials. So what's our motivation to actually look into developing novel antimicrobials? And there was this great paper last year where the authors looked across 88 pathogen drug combinations and found that about 1.3 million deaths were associated directly with resistant bacteria. And they also found that close to 5 million were associated with death, where resistant bacteria were involved. So that's big numbers, and that's for basically each single year. And we urgently need some new strategies, some new developments to treat these resistant bacteria. We basically are a little spoiled over the last decades, grew up with antibiotics that were readily available. And now we're coming to a time point where resistance is quite a big problem now. And what we also see at the same time is that we have this what's often called discovery void, where only very few developments are made in terms of very truly novel antibiotics. So one reason is obviously the overuse of antimicrobials also in veterinarian medicine that leads to all these resistance development. But what's with the discovery void? It is not that we really totally lack new ideas or new substances. I think one of the reasons, I mean, it's a really complex topic, but one of the reasons is also that developing a new antimicrobial is, or from a business perspective, not so attractive because traditionally these antibiotics were really cheap. And we have a lot that they're still used. So introducing a new compound is a challenge because it goes into a highly competitive market that has a low profit range. So, and at the same time, developing a drug costs a lot of money. And despite this 100 years of experience in antimicrobial development, there is still quite a high failure rate when you look what happens between the clinical trials here in phase 1, 2, and 3, and what actually is then really successful in terms of a new drug application. In addition to that, we have a problem that when a company actually does all the required tests and trials and get an FDA approval, it spends maybe 100 million or more. And because of the pricing and the competitiveness of the market, it's sometimes tough to really get a reimbursement in a way, or to get the money back by the sales. And so some of these companies actually get bankrupt or get sold, and then they stop producing antibiotics. And that happens a couple of times. And so now there's ideas of reimbursements from government sides or from other institutions that different governments may set up. So there is development in that area, which makes it hopefully more attractive, again, to produce antibiotics. Now I would like to introduce you to the Cardionic antimicrobial peptides. I just want to say, yes, there are anionic ones as well, but we focusing on cardionic ones, and so far there are way more cardionic antimicrobial peptides discovered as anionic ones. So therefore the focus here is on cardionic ones. So they're basically in any kind of living system, and we know more than 3,000 naturals, and people have obviously created artificial ones, new ones, variants, that we have now 15,000 of those. In higher organisms, interestingly, they are part of the innate immunity, and they're between 12 and 80 amino acids long. As I said, cardionic, amphiphilic, and they do have different structures and functions, which I will show you in the next slides. So there are multiple functions, and as the name indicates, they are antibacterial. And what makes them really interesting for drug development is they are also active against multi-drug resistant bacteria. They can also be active against fungi, which we also have in our screening procedures, and they can be antiviral. We have databases with antiviral peptides, and a part of them coming from these antimicrobial peptides. We have tested some peptides in collaboration with colleagues for SARS-CoV-2 and found 2 that were decently active. So there is definitely a high potential for those activities as well. They can be antiparasitical, especially lots of work was done on malaria. And as I mentioned before, some of these antimicrobial peptides show a good activity against cancer cells. And in higher organisms, they have immunomodulatory and chemotactic functions, and they can really influence the immune system and chemokine and cytokine productions specifically. So that makes them really interesting from the point of drug development with different targets. And structure-wise, we have the classical alpha helical structures, but we also got beta sheet formations, and then we got these combined structures. We have all different structural elements, and often they are held together by cysteine bridges. And then there are some more exotic structures, like these open boat structures, for example, from indolysine. They not only have these different structures, sequences, and functions, they also have different targets. And that makes them, from a drug development point, really interesting, because you can develop various different peptides without having any problem of cross resistance or competing for the same market, basically. Also, I want to mention that some of these peptides have not only one, but different targets. And therefore, it makes it even more difficult for the bacteria to come up with a strategy to be resistant. So how do we synthesize these peptides? So Ronald Frank has developed a method, it's called the spot synthesis, where the peptides will be basically spotted onto a cellulose membrane. And this is now a more or less automated system. And you can see here, this is these cellulose membranes in these 4 frames. And in the scale that we do the synthesis, we get about 600 peptides in one single synthesis. Whereas if you only need binding, for example, and you need lower amounts of peptides, you can do up to 3,000 on these membranes. So once you have synthesized them, you basically need to punch them out, put them in these microtiter plates, dissolve them, and then you have these peptides ready to go in high throughput assays in a 96-well format. If you're interested, I have published this paper describing step by step the method of this spot synthesis. So once these peptides are done, you basically transform them or transfer them into these microtiter plates, as I have shown you, incubate them with your target strains, and then incubate them and do live dead stain and then analyze the data. The same you can do with red blood cells as a really easy way to find out if they are really, or as a first indication, if they are actually toxic or not. So then we have applied this technology, and I was the first person to really apply the spot synthesis technology towards the antimicrobial peptides and cutting a large amount of peptides and investigating them really in detail, how they interact with bacteria. And our first paper, where we used like a natural or a variant of a natural peptides back to A and optimized these peptides towards certain pathogens. Later on, we used all these thousands of peptides that I had synthesized and tested against Pseudomonas aeruginosa, and then really used artificial intelligence to predict novel peptides. And that worked really well. And we also then changed the approach a bit to have a little bit of more easily to understand descriptors to see or understand ourselves how these peptides work. And we also optimized some proline, arginine-rich peptides that are specifically bind on the ribosome. And I also designed some hybrid peptides between these peptides that bind to the ribosome and some peptides where we think has some more different targets in the cell. Also did lipidation and glycosylation, and also used a scaffold to put antimicrobial peptides in the scaffold to make them more stable. So now, after many years, we have discovered hundreds of peptides with high therapeutic potential. And so my opinion was that the peptides that should be moving towards clinical trials are the ones that have a really distinct difference in the mode of action as the conventional antibiotics currently used. However, at the time, there wasn't any high throughput screening system to say, oh, this peptide has a different mode of action as that peptide, and this is also different from the antibiotics. So now we wanted to approach that and develop this new tech solution, basically. And so Axel Rosenhahn is basically an expert in BioSAXS. And we talked about this problem, and we thought it could be a solution, but it would require a lot of pioneering work because nobody had done it before. On the plus side, and the measurements are very fast. You need 2 seconds. You get a robust statistics because you measure over millions of cells 20 times. And so you can, within a few days of time, screen hundreds of compounds. And Ralf Mikut, I mentioned him before, from the KIT, is used the data and analyzed it in a way that we could visualize it, and used the principal component analysis for that. So BioSAXS is currently done on synchrotron, but hopefully soon there will be some technologies that X-ray beams in a lab can be precise and powerful to actually do that. You need longer exposure times, but you still may get good quality results. However, that's in the future. Now we're talking about what we did in the past. So our first approach was we needed a proof of principle that it actually works. And that's the paper. It's finally published after years of trial and error. So what we wanted to do was using different known antibiotics with known mode of action or targets, and then see if we can actually distinguish them using the BioSAXS. So we used antibiotics that are in the cell, acting on the cell wall, or having RNA synthesis inhibitor, or the classical ribosomal ones, and then mixed them with E. coli, and then measured them in BioSAXS, but also parallel with electron microscopy. So the principle is basically that you use, in our case, for the first proof of principle, we used E. coli, added these different antibiotics with different modes of actions, put them in basically an autosampler that then goes into the beamline, and we measure the diffraction at a small angle. So this is the diffraction curves that we see. That's more or less what the original data looks like. And we then need to somehow visualize them, analyze them. And as I mentioned before, Ralf was using a principal component analysis to do that. So in the middle here, you can see the principal component analysis, and you use the linear coefficient 1/2. And so you got this 2-dimensional plot. And I just want to walk you through. So this is basically untreated E. coli, as we used a normal K12 strain from the lab. And this is the EM here around, and you can see pictures here in the PCA1/PCA2. You find it around 0, 0. Now, when we look here, for example, tetracycline and chloramphenicol, they both have this condensed nucleoid, very different from the untreated one. And you can see both of them are distinguishable from these untreated E. coli. So then we have rifampicin, for example, a little bit kanamycin. Here, I think you see it better because you see the nucleoid is actually more extended, like larger than in the untreated variant here. And you can see it lays indeed opposite of the ones that are condensed. Polymyxin B, I think, is well known. Targets basically is the cell wall and the membrane, and it really destroys these structures. And you can see it's really far away from the antibiotics. And the peptide that we used with unknown mode of action, you can see that compared to polymyxin B, it doesn't have the damaged cell wall, so it has still a very smooth surface and the membrane is intact. However, you can see there is definitely changes that are really different from those of the antimicrobial antibiotics that we tested here. And it also ends up to be at this one. You can also see that cefampim here is actually more or less non-distinguishable in that BioSAXS, but you can also see it's really tough to see any difference between here as well. So we also then did time and concentration depending series, and then you can actually optimize the way it distinguishes between the untreated and treated one, because some antibiotics or novel antimicrobials may need longer time, or they're really fast acting. And concentration-wise, you can also see at MIC level or higher MIC level what actually happens. So that was a really great moment to see that, yes, it has potential to be used in terms of classifying the mode of action. And in the next study we did, we tried to really look at what structures may have been influenced in order to get a better understanding about modes of action. And here you see Andreas, who has done a lot of work in optimizing, and then Marcel for measurements of the BioSAXS, and also Axel again. So here the idea was to adapt, or not adapt, but to calculate a really simple model towards the measured data. So basically, Andreas was using a cylinder, a simple cylinder, as the shape for E. coli, and then he had to add some content in it, and then recalculate what that curve would look like, and compare this to the actually to the treated one, or to the measured one, sorry. So we find out that we need the outer shape, which is the cylinder, basically the E. coli, and then we need sizes in order to receive the data that we measured, which interestingly pops up as proteins in DNA and ribosome in the volume and sizes. So that means that we can then look on these three different sizes, what happens. And here you can see, for example, this is plotted in terms of the size for proteins. And you can see there is some changes compared with the untreated control. And you can see rifampicin is clearly distinguished from chloramphenicol and tetracycline, which these two are working more on the ribosome, and this is an RNA inhibitor. The same happens here. You remember that when we look at the DNA, the nucleoid was really compressed with chloramphenicol and tetracycline. You can see that here, whereas it was expanded with rifampicin. So you basically can get an idea of what the mode of action could be. So we have only tested E. coli so far. And then in the next experiment, which was again Andreas, but also PhD student of mine, Martin, and Christoph, it's a postdoc in Axel's lab. So Martin was developing peptides against MRSA, multi-drug-resistant Staphylococcus aureus. And we wanted to see if these peptides kill gram-positive as gram-negatives in a similar way, because what we found in the end was that, yes, we could find superior active peptides against MRSA, but they seemed also be able to kill gram-negatives, maybe not at the same concentration, but they were still active. So we thought that maybe BioSAXS can help with that. And this is again what the curves look like. And you can see here already with bare eyes, and that you can see this green one near MRSA, a gram-positive one, compared to this reddish line here is E. coli, gram-negative. You can see they're very different. And they should be, because first of all, one is gram-positive, one is gram-negative. They have a different structure on the cell wall, and they also have a different geometry, where E. coli is more cylindric, MRSA, Staphylococcus is more a sphere. And you can see that when we treat them with peptides, they become more similar to each other. And that's what we basically see also in the PCA. We have here gram-positive MRSA, gram-negative E. coli. And when we treat them with the with the peptides, you can see they actually come closer together. So our understanding is that the mode of action in both of these organisms is very similar, because it leads to very similar structural changes. And we also did EM on them, and you could see that this is the E. coli, and you can also see again this little bit more condensed nucleoid, where for staph it is very difficult to see the effects. However, with the help of BioSAXS, you can see without a problem that they do become similar, more similar in terms of structure, internal structures. Sorry. And so our very first application was to find out if designed hybrid peptides that I told you earlier about, that are combination of arginine proline rich peptides that bind to the ribosome and some small peptides that we developed and having a very different mode of action. Are they, when we combine them, do they actually tend to go in one mode of action or the other mode of action, or do they actually do both and have a combined effect? So that was the question. And as I was saying, we used proline arginine rich peptides, and that was a collaboration with Marco Scocci and Daniel Wilson. And Daniel resolved the structures of the peptides on the ribosome, and I created these hybrid peptides. You can see here these short optimized peptides that we had in the lab, and then we combined them with these optimized proline arginine rich peptides. So we did again BioSAXS. You can see the original curves again, and now the PCA. You can see this is here the untreated version, and that is the proline arginine rich peptide that binds to the ribosome. And here you can see our small peptides that we had optimized, very different. And now the hybrid peptide seems to be really working on both targets. And I also designed a hybrid peptide that falls apart when it enters the cell, and then it becomes closer to the parent here, which makes sense because it's the more active peptide. That means once it is cleaved in the cell, in the bacteria cell, it then needs less concentration as the proline arginine rich peptides to kill the bacteria. So for us, that makes total sense. And then we also looked if some other antibiotics will have a similar mode of action, and you can see they are apart, which is really good, especially here our hybrid peptide is very far apart and could move forward in a way that, because it has two modes of action combined in one molecule, and that makes it very difficult for a bacteria to escape that. So now one question we had, because we are working on these antifungal peptides, is can we do this actually with Candida, with the fungi? And so now the problem here is that we've only measured prokaryotic cells, and they are not that structured compared to a eukaryotic cell. So the big question was, when a substance changes some ultrastructures here, we can obviously measure that. But when something changes in here and most of these structures stay as they are, can we then actually see these changes, or is it actually then hidden in the noise? And so this is our last project, and we used flucytosine as an antifungal compound, and also some peptides that we had in the lab that are antifungal, and then measured that. And we are basically having submitted a manuscript, and we're showing that indeed it works. And it could basically mean, yes, you can not only test your antibacterial, but also your antifungal compounds with that. So now I would like to come to the take-home message. So traditionally, you would do a whole cell antimicrobial screen, use cytotoxicity screen to select your compounds. However, that is a risky route because you can run into the same mode of action as conventional antibiotics, and then your novel compound is faster off the market as it was on. And in order to de-risk it and enrich really compounds that have novel modes of action, you can do your same screening, but add BioSAXS to your compounds that, your best 100, 200, if you have large amounts. And I have talked all about peptides now because this is our specialty. But the method of BioSAXS really has no restrictions to peptides. It can cope with any kind of antimicrobial substance you develop. And therefore, it's really exciting. And I hope something interesting for you to consider in your projects. With that, I would like to thank all the people involved, and I would like to thank you for your attention. And then I guess we can have some questions now.
Thank you so much for your presentation, Dr. Hilpert. It's now time for our live Q&A with the speaker. To ask a question, just type it in where it says questions and then press submit. And I see we've already got a few, so thanks for those who have already submitted questions. All right, so to get started, and this question ties in well with how you ended up your presentation, but someone was wondering about how BioSAXS can be used for other antimicrobial compounds and classes. Do you have some examples of different things that it could be used for?
Yeah, so the beauty of this method basically is really that there is not really a limitation of what you can do. So we, for example, have developed some antimicrobial lipids, for example, that you can also easily measure there. And so I know that, there's basically more or less daily a paper describing a new antimicrobial compound of some sorts. So, and often the question is it actually worthwhile to proceed? And that is a big question, because if you really want to answer that, you need to take into account the mode of action and toxicity, of course. But mode of action can cost you quite a lot of time and money. So if you can stick it for 2 seconds in the system, you can actually do quite a bit. Testing various variants and whatever. There's no restriction really. And that's why I find it so exciting to share this new option or method, because I hope that many people will make use of it.
Yeah, speedy data is always nice. All right. Oh yeah, that actually ties in perfectly for our next question, which is asking whether the measurement really only takes 2 seconds per sample.
Yeah, in principle, the time the sample spends in the beam is indeed 2 seconds, more or less. However, obviously there is an autosampler. You have to load the autosampler, and then the sampler goes in and goes out and goes in. So there is a little bit of delay, but it's quite amazing that in these 2 seconds, you basically measure your sample with millions of cells 20 times over. It's just like not one beam, it's like 20 times. And so you get quite a lot of data out of that. And so it's always a good thing to have a really robust statistics. If you think for your alternative, basically, let's say you make a microscopy, as I showed you, EM, it takes a very long time to prepare all the samples and then to measure them. And imagine you would analyze millions of cells 20 times over to get the same statistic. It would take year to kind of measure that. So yes, it's quite astonishing that it takes so little time.
In terms of sample preparation, how much volume do you use for each sample?
So currently, we're using in our preparation, we use normally 1 milliliter or 2 for let the bacteria grow, and depends a bit on the density and on the bacteria, how well they form pellets. So you can adapt that. But then when you do the experiments in itself, you have around 200 microliters of volume. And I think it's around 100 microliters that actually get used to have the beam through it.
Gotcha. Perfect. Someone else was wondering if you perform time and concentration dependency studies.
Yes. So after we have done the first proof of principle, we also included some other peptides that we didn't know the mode of action. And we compared classic, all the different groups of antibiotics that are around, and the peptides. And we did time and concentration series. And that was quite interesting because you can clearly see that some of these peptides, they have done their job basically in 40 minutes. Whereas some of these antibiotics, until you really can measure the ultrastructural changes, it takes sometimes 10, 15 hours. So, and that makes the direct comparison a bit tricky, right? Because if you say, oh, this antibiotics is here and this is there, you do have to check some timings. So what we normally do is we do these classical time kill experiment where you look killing events over time, and then you get a clear idea of how fast they are. And from that information, you then design time points for your measurements. And the same you can do with concentration effects. It is interesting, obviously, to see, if some antibiotics do tend to have a secondary effect if the concentration really goes up a lot. And you can see that in this method as well.
Another one of our audience members was wondering about whether there was an application of the technique to antiviral drug discovery.
As far as I know, the answer is no. But I cannot really be 100% sure since I have never looked in antivirals in with that method. However, there are obviously antiviral peptides, and the limitations is basically if you have a virus and you have an antiviral compound, thus the compound changes somehow the structure, because the virus is compared, to a bacteria or to a yeast is non-reactive in a way that it doesn't have its own metabolism. It doesn't react in a way a cell would. So if you do have an antiviral drug in mind, have a think about, will it change somehow my virus? Will I expect some changes? Does it bind somewhere specifically? Does it, I don't know, destroy the capsid or the lipids around it? And then yes, you can definitely measure it. Obviously, then it depends also on the size of your virus, but it shouldn't be really a problem.
Have you had the opportunity to look specifically at cell membrane targeting pathways of any AMPs?
Well, in our first experiment, we used polymyxin B, which is the classical membrane targeting example. And we can clearly see the effect and differences. It's quite dramatic to everything else we have measured so far. Sometimes it's a bit tricky if they are really highly lytic, because then if they're really, really fast, it's really tough to actually get a pellet anymore, because you can imagine if the bacteria has like a certain shape and then it leaks out and all these stuff floating around, it breaks off, and then you don't get these structures anymore. So there, the timing, I think, and the concentration might be extremely important to look at.
Another audience member was wondering if you've done any screening against proteins using your libraries.
Screening against proteins. I'm not quite sure. Maybe he or she means that we have looked to find binders to certain bacterial proteins. I'm not quite sure. If yes, then you can also look in BioSAXS if the structure changes. That's quite I would say a more classical approach for BioSAXS. People have done that before many times. So yes, and then you can compare it also to live bacteria. So that would be possible, but maybe I just misunderstood the question.
I'm not sure. Can BioSAXS see the action of peptides that only cause pore formation with no intracellular target?
Well, that's a tough one. So if you get pore formation, you normally would see depolarization of the membrane, and you would most likely see some kind of smaller contents leak out. That would lead to definitely a strong reaction of the bacteria. So you may not see the pores in itself, but you actually may see the resulting effects that the bacteria tries to overcome these problems. Yeah.
Okay, interesting. How do you intend to scale up peptide production to get the best candidates into the market?
Scale up peptides. Well, I think the question is about what method we would use to produce large amounts of peptides, because spot synthesis is obviously not for large amounts of peptides. So if you use peptides, I mean, the classical one you would go, let's say in clinical trials, you would definitely use chemical synthesis. And luckily, the field actually moves to a bit more of a greener synthesis. So from that point of view, you can have really ultra clean peptides for your clinical trials. Once you need more, let's say you need a ton scale, then alternatively, yes, there could be production in E. coli or some other system. Yeah, that would be my answer.
Gotcha. We got a note of clarification on the previous question. So they were asking about directly against protein identified as targets. I'm not sure if that helps or not.
Oh, if you can use directly against protein identified as targets. Protein against. So they have identified proteins that are targets from some drug, or they want to develop some drug against those targets, I assume. then yes. As long as there is a reaction on the size of changes in structure or a response of the bacteria, then yes, you would see that. you may not really, you obviously cannot say that I can see that protein and it changes its structure. That's the method is not, that of a solution or a high resolution that could do that. But you can definitely say, oh, I have this drug, it does interact with this protein. Then yes, you can definitely see if there is a change and what that looks like. Yeah.
All right, I think we have time for a few more. Someone was wondering, have you used this approach with blood cells to check hemolytic properties to see if they are toxic?
So basically, screening, we do screen when we, well, I started new. When we do screening projects, we synthesize 600 to 3,000 peptides and then screen them. And so when we do screen them, we do against our targets, which might be, E. coli, Pseudomonas, or something else, a fungi. And at the same time, we also do an hemolytic screen in parallel, or say, with the same batch that we produce.
Mm-hmm.
So we do have already this information. So we don't really need to go in and repeat this in the BioSAXS. You still have to imagine the time is still quite valuable because you go on a synchrotron. You want to really measure things that you can't otherwise measure.
Right. Can you use this for screening a deep mutant library of AMPs expressed within the bacteria?
Can you say again? Can I screen?
They were wondering, can you use this for screening a deep mutant library of AMPs expressed within the bacteria?
Yeah, I think you could. I mean, it depends a bit on the. I mean, there's just so many different effects a mutant could show. Basically from not even growing to have no effect, and everything in between. So if you get a structural change in your bacteria that is strong enough to pick it up, or it causes some changes in the bacteria because of the stress. I mean, if you express, for example, something and E. coli doesn't like it, it often puts it into inclusion bodies. You would see that. If you have lots of misfolded proteins, let's say you mutate an chaperone or a protease of sorts that is needed, and it's not there, it doesn't function, you would then see the stream down effects, basically, or downstream effects, sorry, on the structural side. So maybe like a yes and a no. You would see changes if they occur on a structural level, but if the changes are too little, then you would miss it.
I see. All right, and one more. Someone was wondering if you can distinguish between bacteriostatic and bactericidal antibiotics, since you did a time course.
Yes. It's interesting you asked. We have done these studies, as I said, and yes, we could actually say that it's bacteriostatic versus killing. However, we have never really repeated those experiments enough time that I, be really comfortable to say yes. So we have done an experiment, not only one, but we have done basically 3 repeats of that experiment, and it showed yes, you can do that. However, we haven't tested it in any different setup, which I'm always kind of cautious to say, oh yes, it works.
Yes, that makes sense. Well, unfortunately, I think that's all of the time we have for today. Thank you so much, Dr. Hilpert, for your wonderful presentation and for answering everyone's questions. And a huge thank you to the audience for being here with us today. I would like to kindly invite all of you to take a few minutes after the webcast ends to just answer a brief survey that will be launched. And please remember that you can always watch this webcast again at any time on demand at nature.com webcast. Thanks so much for watching, and I hope you'll join us again soon.
Thank you all.